Okay, so picking up where we left off last time, got to finish talking about production of saliva from our salivary glands. What's all in saliva? We're going to talk about digestion with regards to the mouth. Two forms of digestion take place thanks to our mouth: mechanical, chemical. Mechanical, as you guys learned last time, is a form of physical breakdown where you're taking something bigger, the food, and making it smaller. And this is the primary place where we perform mechanical digestion. We call this chewing. Okay? Most commonly called chewing. Fancy term for chewing is mastication, where we manipulate our food using our tongue, grinding it with our teeth, and mix up that food with the saliva, forming what's called a bolus. A bolus is just a fancy word for a soft, flexible, easily swallowed mass. And, along with mechanical digestion, we're going to perform a kind of more complex form of digestion, chemical digestion. Remember, chemical digestion involves chemical breakdown. We're breaking bonds using enzymes. Two enzymes I want to talk about produced in our mouth digest two different things. Salivary amylase is produced by our salivary glands that acts on starches, which we know as polysaccharides, are a complex form of carbohydrates. In fact, most of the carbs that we eat should be this type. Polysaccharides are not able to be absorbed, nor are disaccharides. Polysaccharides, remember, are long chains of monosaccharides. Monosaccharides, if you think back to that biochem lecture, would be like glucose and fructose. Small, 6 carbon or so molecules, which are the building blocks for making our disaccharides and our polysaccharides. So we start digesting those in our mouth. Now, salivary amylase is a protein. It's an enzyme, and that makes it a protein. And thinking back to the beginning of the semester when we talked about proteins, we talked about how proteins are sensitive to their environment, and I used the slinky analogy, how proteins have a specific shape that affects their function, but if they lose that shape or they unwind, they lose that function. That's what we call denaturing. And that's what can happen to salivary amylase, and that's what actually happens to it when it arrives in our stomach, because of the stomach acid that we produce in our stomach is very low in pH compared to the pH of our mouth. Now, it's not an instant deactivation of salivary amylase, because it takes a little while for that salivary amylase to come into contact with the stomach acid, but in a fairly short amount of time, the salivary amylase stops working. So it's not a great enzyme, but it does get the ball rolling for the digestion of starches or fat or carbohydrates. Excuse me. Speaking of fats, our first fat digesting enzyme is also produced in our mouth by the lingual glands of our tongue. Lingual lipase, produced by our tongue, acts on the primary form of fat that we have or ingest in our body, triglycerides. Acts kind of the opposite of salivary amylase in that it is not active until the acidic environment activates it. The acidic environment of our stomach activates it. Okay, so what I'm going to do, we're going to have a pretty good tally of things for digesting our carbs. We'll have a tally for fats. We'll have one for proteins. So we'll start with our first, which is salivary amylase would be our first carb digesting enzyme. Our first fat digesting enzyme will be lingual lipase. So we'll refer back to this at various points. We'll move on to talk more about enzymes here a little bit, because our pharynx and the esophagus do not produce any enzymes. Pharynx and the esophagus are going to be involved, though, with something very important, which is swallowing. Okay? When food is first swallowed, it passes from our mouth into the pharynx, which is a funnel-shaped tube, which extends from the internal nares to the esophagus posteriorly and the larynx anteriorly. It's actually a shared tube. We didn't really talk about it with our respiratory system, but it does function for respiration, for breathing, because it carries air from our nose and from our mouth to the larynx, but it also is what carries our food and drinks into the esophagus. Composed of skeletal muscle, which we should know by now is voluntarily controlled, and that's important because that's something we'll talk about regarding swallowing. 3 parts of the pharynx exist: a naso, an oro, and a laryngopharynx. As far as the nasopharynx, it really is not important to us in this lecture because you're not ingesting food through your nose, you're not drinking water through your nose, you're breathing through your nose though, and nasopharynx is only functioning in respiration. The oropharynx functions in digestive and respiratory functions because this is the second half posterior to the mouth that carries food and air up and down through the oropharynx. Stuff goes down into the laryngopharynx, and I say stuff because it could be food and drink, or it could be air, so it's both digestive and respiratory in function. Since we've got skeletal muscle in the pharynx, it's very involved with the kind of active voluntary aspects of swallowing, pushing that food, that bolus as we can call it, from the mouth into the oro and then laryngopharynx, finally into the esophagus, which is another tube, much longer, about 25 centimeters or 10 inches in length, that runs behind the trachea and just is really just a straight up food tube. Has no other role other than just carrying that bolus into the stomach. As you can see, it does not have any enzymes or perform any absorption. Mucus that's secreted here helps with lubricating that food, helping to make it slide down more easily, and it's produced and released by the mucosa of the esophagus. Now, we talked about in an earlier slide about the 4 layers: mucosa, submucosa, serosa, muscularis. Our esophagus has those 4 layers. I'm not going to talk about all of them. I do want to talk about just 2 of them. Mucosa has some important features of the esophagus. It is, aside from producing that mucus I just mentioned, it's pretty resistant to wear and tear because we don't swallow something all the time that's nice and soft. We sometimes swallow stuff that's really hot. Sometimes it's kind of jaggedy, and it's pretty resistant to those abrasive substances that it comes into contact with. Okay. The muscularis is actually divided into thirds with the esophagus. The superior third is made up of skeletal muscle, so its contractions are under voluntary control, which is helpful during swallowing. Middle third is skeletal and smooth muscle, and then the inferior is strictly smooth, which is, as we know, completely involuntarily controlled. There's a sphincter at the top and a sphincter at the bottom of the esophagus. A sphincter is a ring of muscle that, in this case, controls the movement of that food into for the upper and then out of for the lower esophageal sphincter. The lower esophageal sphincter is located at the junction between the esophagus and the stomach, and is going to prevent contents of the stomach from spilling back up into the esophagus. Unfortunately, that doesn't always take place, which we call that condition where a person experiences the burning in their esophagus. You kind of commonly call it heartburn, but it's more sophisticatedly known as GERD, gastroesophageal reflux disease. And it's very painful and unpleasant because of the acid that our stomach has, and the juice of our stomach. Our stomach's really good at being resistant to that because it produces a really thick layer of mucus. The esophagus doesn't have it as much, and it's more resistant or less resistant as a result. Now, when we swallow our food, gravity is definitely going to help play a role with it, because typically when we eat and when we drink, we are sitting in an upright position. But there's kind of also a very important wave-like contraction of muscle that helps push that food bolus downward. We call this peristalsis. And we'll talk about that here on this page. Peristalsis is one of the 2, segmentation being the other, forms of GI motility. We learned earlier in the lecture lecture that motility is just the capability of our tract to move stuff along the length of it, thanks to the contractions of the muscle within the wall of the tract. Peristalsis is a key, or the key with peristalsis is that it's going to push food forward only. And it's because contractions are taking place behind, and it's like squeezing toothpaste from the back end of the toothpaste tube on the opposite side of the cap where the toothpaste comes out, pushing it always towards that opening. Okay? And this is what we do, starting with our esophagus and continue it all the way down through the anus. This is the primary movement and the way that we get food all the way out, from one end to the other. So it's not just gravity helping us swallow, it's peristalsis as well. Okay. Segmentation, however, is actually a form of physical mechanical digestion because it is going to cause food to go both forward and backward because there's contractions taking place at various segments of the tract. And basically, it's an alternation between contraction and relaxation, like this segment here, which is relaxed, and then here later on, it's contracted and it's relaxed again. But it creates a contraction that pushes the food in opposite directions, forward and backwards, making them, chopping them into small pieces. So it helps break down the food particles, mix them up with the digestive juices, and bring the digested food into contact with the intestinal mucosa to facilitate absorption. This takes place in our small intestine in response to distension and involves contractions of the circular muscle fibers of the muscularis. So instead of grabbing the toothpaste tube from the back end of it, it's like grabbing it in the middle, squeezing. You're going to end up pushing toothpaste in opposite directions, which is one way to squeeze the toothpaste, not the way I do it though. Okay, I've been mentioning about swallowing here for a couple of slides. We'll talk about that right now. The fancy term for swallowing is deglutition, and it's not just the act of moving food from our mouth into the throat. It actually is from the mouth all the way down to the stomach. And it's facilitated by secretions of saliva and mucus to help lubricate the food, forming that bolus and then helping it slide more easily. There's 3 stages. The first is listed there on the previous page as an oral stage. Here on this next page, it's called a voluntary stage, and that's because it's the only one that we have direct control over, where we can get that bolus from our mouth into the oropharynx. The second stage and the third stage, as you can see, are completely involuntary. The second stage is going to involve the pharynx, which is why it's called the pharyngeal stage, moving the food from the pharynx into the esophagus. The arrival of a bolus into the oropharynx is going to stimulate receptors that line the oropharynx or within the wall of the oropharynx, causing signals to be sent up to the deglutition center, our swallowing center, which is in the medulla oblongata and our lower pons. As a result, signals come down, causing kind of a couple things. Our soft palate, the roof of our mouth, towards the back of our mouth, and the little ball that hangs down called the uvula will push upward and close off any bit of food or drink from entering the nasal cavity. Okay. It'll also cause the epiglottis, this little flap here, to go from a open stage to a closed stage. Prevents you from choking, prevents any food or drink from entering our larynx. Okay. So that's what all this was. I just showed you inside of Reddit. The esophageal stage is the final stage, another involuntary stage where we pass that bolus from the esophagus into the stomach. It involves that peristalsis that I just talked about due to the contraction of the circular and longitudinal layers of the muscularis. Mucus secreted by the esophageal glands lubricates the bolus and reduces friction. Passage of solid or semi-solid food from our mouth to the stomach takes about 4 to 8 seconds. So it's not an instant thing, but it's not a super long-term thing. Your very soft foods, liquids, are going to take just about a second, so not a whole lot of time. Peristaltic contractions are so strong in our esophagus that you could actually do a handstand after or even while standing, and food would still actually reach your stomach. So kind of cool. You can actually be inverted and still get that food down into your stomach, which I don't really recommend. It's kind of stupid to do, but you can do it. Okay, moving on to the stomach, a portion of our digestive tube that will dilate or expand into an elongated pouch-like structure. It is able to do that because on the inside of the stomach, you can kind of see them here, it has these accordion-like folds called rugae. Think like an accordion has the ability to kind of stretch out. That's what our stomach has the capability of doing. Anytime you see the word gastric, it refers to the stomach. You see gastric kind of all over the place on this page. First gastric thing I want to talk about are the gastric glands, which are found throughout the wall of our stomach and channel their secretions of their cells along the ducts into the lumen, into the space of the stomach. Several of those gastric glands and cells open into the bottom of narrow channels of what we call gastric pits. Gastric pits are exactly what it sounds like, little depressions or pits within the wall of the stomach, within most notably the mucosa. We zoom in at a gastric pit like we do right here. You see there are several different types of cells found within it, and I want to talk about those on this page, or right here on this page. Okay. First two, I kind of lump them together. Both of them do the same thing. They secrete mucus. The difference is based on, or their name anyway, is based on where they're located along the pit. The surface ones are located more towards the surface, pretty common sense. Neck cells down here are farther down in the pit. Both of them do the same thing, secrete mucus, which, as I mentioned, is very, very important for protecting the stomach from that hydrochloric acid that I've referenced. Speaking of hydrochloric acid, produced by parietal cells located in the pit. I'll talk about what hydrochloric acid does here in a little bit for us. There's also a chemical that it releases called intrinsic factor, which is needed to absorb vitamin B12. Another cell called chief cells, shown here in purple, secrete two different enzymes. Actually, it releases pepsinogen, which then is converted because it's a precursor to an enzyme pepsin. Pepsins are only protein digesting enzyme in our stomach. It'll be our first one to add on the protein list. It is very active in our stomach, just like lingual lipase I mentioned, because of the acidity caused by that hydrochloric acid. Chief cells also secrete another enzyme, gastric lipase, which helps split short-chain triglycerides into fatty acids and monoglycerides, so it helps in breaking down fats. So I'll flip over here and I'll add gastric lipase to the list of fats. And then I will add, for the first time, something to proteins, and that is pepsin. Okay, other things. Other cells release histamine, serotonin, two chemicals that regulate aspects of digestion produced, you can't see them here, but they're produced by enterochromaffin-like cells. G cells secrete gastrin, which is actually a hormone, which means that the chemical is not going to go into the lumen of the stomach. It's going to go into the bloodstream. Gastrin and histamine help stimulate the release of that hydrochloric acid from the parietal cells. And then there's another hormone called ghrelin, released by gastric cells. Also, you can't see them here. Gastrin is released in the bloodstream to promote hunger when our stomach is empty. Now, our stomach is very involved with both chemical and mechanical digestion, but we don't do a whole lot of absorption of nutrients in our stomach because the epithelial cells are pretty impermeable to most materials. However, those mucus cells of the stomach can absorb some water, ions, some short-chain fatty acids, and even some drugs and alcohol. And as far as drugs, one of the most notably would be aspirin, which is why, aspirin and alcohol as well, when ingested, really their effects are pretty quick acting. We don't have to get them farther down in the tract to be absorbed. Now, before we move on from the stomach, we got a little bit more to talk about. On this page, I want to talk about what takes place in our stomach with regards to digestion, with mixing up the contents, the food, with the gastric juice that we just talked about. Several minutes after food enters our stomach, waves of peristalsis pass over it for about 15 to 25 seconds, or every 15 to 25 seconds, say. Most of these begin at the body of the stomach, which is this kind of middle area, and then they push forward towards the end of the stomach where that pyloric sphincter is at. They intensify in this area, what we call the antrum, the portion that tapers towards the small intestine. Okay. This forward movement of material is called propulsion. Okay. But at this point, and you can see it here in this figure, the pyloric sphincter is pinched shut, preventing most of the contents from entering the small intestine. So what happens is that wave of contents hits the stomach wall, and like a wave of water, when it hits the wall, it's going to have to flow backwards, and that backwards flow of material is called retropulsion. So what we're doing is we're just rocking the boat here and mixing stuff up in our stomach to form what's called chyme. Now, I'm going to try to call it chyme at this point and not call it food, because really it's not food. It's food plus gastric juice, which is a very significant amount of stuff. Once the food particles in that chyme are small enough, they can then pass through the pyloric sphincter. When the pyloric sphincter relaxes, it doesn't just allow everything out at once. Through the process of what we call gastric emptying, we release a very small amount, and we do so very slowly, which takes about 2 to 6 hours. And that's because we're only emptying about 3 mils at a time. 3 mils is a very insignificant amount. One of my biggest pet peeves is when a person, eats, and then 15, 20, 30 minutes later, they say, "Oh man, that ran right through me." There's absolutely no way that, they've passed that food all the way through them. More than likely, that food is still sitting in their stomach, being digested. So what's actually happening is, when you eat, stuff kind of starts to move along the track as you start to empty your stomach, which causes you to go to the bathroom, pushing out, whatever you ate, maybe, 8 hours, 10 hours before, last night, whatever. The emptying of our stomach is controlled by both hormonal and nervous mechanisms that we'll talk about later in the lecture. During the time when we mix our chyme up, digestion by salivary amylase continues into the acidity of the gastric juice and activates it. Like I said, salivary amylase likes our mouth, not so much our stomach. And that acidity, though, activates the action of the lingual lipase enzymes from our mouth to begin the digestion of triglycerides. So hydrochloric acid is involved with activating lingual lipase enzymes and activating salivary amylase. We learned just previously that it's produced by parietal cells, but it's actually kind of unique in how we produce and release it. Hydrochloric acid, as you should remember from the beginning of the semester, is made up of 2 ions, hydrogen ions and chloride ions. We actually secrete them out into the lumen independently. Both of them are released by parietal cells, but they don't get released together. The hydrogen ions are released independently thanks to a protein pump called hydrogen potassium ATPase pump, which performs active transport, pumping hydrogen ions against their concentration gradient. Since it's active transport, we know that, and we know that it requires ATP. While doing that, potassium is able to come into the parietal cell, so it's kind of an exchange situation taking place. Okay. At the same time, chloride is being pushed out of the parietal cell through a different channel. And then the hydrogen ions, the chloride ions added in the lumen can unite, forming hydrochloric acid. Now, we're continuously pumping out hydrogen ions from our parietal cells because we're continuously producing it inside. Parietal cells have that same enzyme that we talked about with red blood cells called carbonic anhydrase that joins water and CO2 forming carbonic acid, which then, as we know, breaks apart to hydrogen ions and bicarbonate. This is where those hydrogen ions come from. The bicarbonate that's formed will exit the cell, enter the blood, and contribute to pH of our blood, and also get carried to our lungs to be exhaled as CO2. All that stuff we talked about in our last lecture. So more hydrogen ions are formed in the parietal cells due to the action of carbonic anhydrase, combining CO2 and water, forming carbonic acid. Carbonic acid then associates to the hydrogen ions and bicarbonate ions. Bicarb leaves the cell to enter the bloodstream while the hydrogen ions continue to be pumped into the lumen. So why do we secrete hydrochloric acid? Well, these three things will tell us why. Okay. First of all, the proteins in our food are denatured at lower pH, which makes them more digestible. So going to my list, I'm going to add HCl. Keep in mind, hydrochloric acid is not an enzyme, but it denatures, unwinds the proteins that we ingest. Denatured proteins are easier to digest when they're unwound. And so unwound proteins are easier to digest than wound up proteins. The acidity of the stomach helps activate that enzyme, pepsin. I don't list it, but it helps activate the lingual lipase. And as we learned previously, the acidic environment of our stomach helps kill bacteria and other microbes. Secretion by our parietal cells is related to three different things. Acetylcholine, which we know is released from our parasympathetic nervous system neurons whenever we have a rest and digest response. That can be one trigger to release hydrochloric acid. Gastrin, which we just learned, produced by the stomach, acts directly on those parietal cells to trigger the release of HCl. And then histamine, we also learned, released by the cells of the lamina propria of our GI tract, which includes the stomach, triggers the release of HCl. So one from the nervous system, two chemicals actually released by the digestive system itself. Okay, what we're going to do now for the next 3 or 4 slides is actually kind of take a detour away from the tract. We're going to now move into 3 accessory organs: the pancreas, we'll get to the liver, then into the gallbladder. And I talk about those before we move into the small intestine, because the pancreas produces a chemical, a bunch of chemicals really, a fluid we call pancreatic juice. There's bile that we produce by our liver and store in our gallbladder. Those 2 fluids, bile and pancreatic juice, are carried by way of some duct networks and dumped into the duodenum of our small intestine. The duodenum is the beginning of our small intestine. So I wanted to talk about these 3 accessory organs that produce chemicals that are dumped into the small intestine before we talk about the small intestine. Okay, pancreas, as we learned about earlier in the semester, is an organ that produces hormones like glucagon, insulin, produced and released by what we call pancreatic islets, or islets of Langerhans cells. There's only 1% of the cells of our pancreas that are these type, meaning that there's 99% of the cells that are exocrine function. We call these the center cells, and these are the ones that produce that pancreatic juice I was just mentioning, a clear colorless liquid, mostly made up of water, contains some salts, sodium bicarbonate, and several enzymes. The enzymes we'll talk about here in just a second. First, let's talk about that sodium bicarbonate, which is a base and gives pancreatic juice a slight alkaline pH, 7.1 to 8.2. It's very important to help buffer the acidic gastric juice and the chyme. I said earlier that the stomach is really, really well suited for the hydrochloric acid that it produces. It's got a thick layer of mucus protecting it. I mentioned that the esophagus doesn't, your small intestine doesn't either. Well, that hydrochloric acid is going to be in the chyme that's going to be released into the small intestine. And if we didn't have buffers like sodium bicarbonate to neutralize that hydrochloric acid, the hydrochloric acid would basically eat away at the small intestine. So sodium bicarbonate, remember, is a base. When bases and acids come together, they cancel each other out, essentially. Okay, let's talk about the enzymes. First, pancreatic amylase is our second digester of carbohydrates, starches. Remember, those are polysaccharides. Trypsin is another protein digesting enzyme. Pancreatic lipase, as you can see, is listed as our principal triglyceride digesting enzyme in adults. So this is our most important fat digesting enzyme. Phospholipase helps digest phospholipids. I'm going to go ahead and list that on my tally there for fats. When we talk fats in our diet, mainly we're talking about triglycerides, but we do ingest phospholipids, which are another form of fat, and we have to digest them. So I'll list it because I didn't really specify what type of fat. And then a couple other enzymes, ribonuclease, deoxyribonuclease. I'm not going to list those anywhere. They digest nucleic acids. Remember, nucleic acids are the RNA, DNA molecules that we learned about earlier in the semester. Ribonucleic acid is the full name of RNA. That's digested by ribonuclease. DNA is deoxyribonucleic acid, broken down by that enzyme. We don't have a lot of DNA and RNA in our diet, but when we eat things, they are going to have those nucleic acids in them. So they are something I did want to mention, not something I'm going to focus too much on. Okay, listing here things out. Add trypsin to our list for proteins. For fats, we will add pancreatic lipase. And then for carbohydrates, we will add okay. The stimulation of pancreatic juice release is controlled by hormones. Once we have chyme, once again, remember that's food from our stomach that is now starting to fill up the small intestine. So we're releasing it into the small intestine. The arrival of chyme in the small intestine is going to drop pH in the duodenum below 4.5, triggering the release of secretin. Okay, here's something you need to do to keep your head straight. Highlight the fact that this is a chemical, it's a hormone released by our small intestine. Okay. Secretin and cholecystokinin that I'm about to talk about are both released by the small intestine. I list them on this page where I'm talking about the pancreas because they act on the pancreas. Secretin is what stimulates the release of that bicarbonate that I mentioned earlier to help neutralize the acidity of chyme in our duodenum. Cholecystokinin is released by our small intestine when fat and protein is found in the chyme that's arriving at the duodenum. CCK, as it's abbreviated, stimulates the pancreas to produce those different enzymes. So when we have fat and protein in our chyme, the acid from the hydrochloric acid, when those are starting to arrive at the small intestine, the duodenum, we release secretin and CCK from the small intestine to start releasing pancreatic juice. Okay. CCK and secretin also stimulate the liver to produce bile, a chemical that we produce in our liver and store in our gallbladder to help us digest fats and oils. And we'll get into that more here in just a second. Major nerves innervating the pancreas are the vagus nerves, preventing parasympathetic innervation, which, as we know, it's all by now rest and digest. Whenever that's active, so is your digestive organs. Whereas like sympathetic fight or flight responses inhibit the secretion of, pancreatic juice in this case, because we need to reroute blood flow to the working tissues like our skeletal muscle as we fight or flight. Okay, the liver is our largest gland in the body, weighing in at approximately 1.4 kilograms or 3 pounds, made up of 2 lobes, which then can be divided into smaller, what we call lobules. 2 methods of delivering blood flow to the liver exist. We have what we call a hepatic artery that delivers arterial blood, but kind of more importantly, for digestive reasons, is nutrient-rich blood coming by way of what we call a hepatic portal vein. I call it nutrient-rich blood because it's going to contain nutrients like the carbs, the proteins, the fats that we just absorbed in places like our small intestine and our large intestine. A portal vein is a type of vessel that can carry blood from one organ to another. In this case, several organs directly to the liver without having to pass that blood to the heart first. Okay. So what this enables for is those nutrients, carbs, proteins, and fats to go basically directly to the liver before they go anywhere else. One of the main functions of your liver is to produce what we call bile, about a liter of it a day, 800 to 1,000 mils. Bile is a yellow, brownish, or olive green liquid. pH is 7.6 to 8.6 and is made mostly up of water, bile salts, cholesterol, lecithin, bile pigments, and several ions. It's partially an excretory product. Okay, back up, sorry. Okay, so I was saying it's partially excretory, which means it's going to be pushed out of our body, and it's going to be a component of feces. Partially digestive in function, okay, for its role in what we call emulsification, using those bile salts. Okay. Emulsification, I can just best show you with this picture, is another form of mechanical digestion where we take fat globules that are very big and we break them down into smaller fat globules. Okay. And we have to do this, and this happens because, as you guys remember from when we first learned about fats and water don't get along. There's going to be a lot of water in our stomach, whether that's just from us drinking it or as part of the gastric juice. And since the fats don't like water, all the fats are basically going to just come together and create just big old glob. And a big glob of fat is going to be really hard to digest. So what these bile salts do from the bile is they come in and they chop up those big fat globules into smaller ones. And those smaller lipid globules are going to be a lot easier to digest. And so that's what emulsification is. So I'm actually going to add under pancreatic lipase bile. Okay. I'm going to put in parentheses, it's not an enzyme, kind of like hydrochloric acid is not an enzyme, but it's very important for emulsification. Principal bile pigment is bilirubin. We've talked actually about bilirubin before. It's formed from the cycle parts of hemoglobin. One of the functions of our liver actually is to recycle parts of hemoglobin. Aside from secreting bile, you can see there are a lot of other things our liver does. It metabolizes our 3 fuel nutrients that we've been kind of keeping track of. And we've talked about really, especially these first 2 before. Okay. It's involved with maintaining blood glucose levels, blood sugar levels, by either releasing glucose into the blood or taking it out of the blood. If you think back to our lecture on the endocrine system, we talked about 6 processes. I wrote them down or typed them out. You had glycolysis, glycogenolysis, glycogenesis, gluconeogenesis, lipolysis, lipogenesis. Okay. Our liver can do a lot of this. And when our blood glucose levels are low, it can break down glycogen to glucose. That's glycogenolysis, releasing that glucose into the bloodstream. It can also can convert amino acids and lactic acid along with other sugars like fructose and galactose into glucose. Amino acids and lactic acid made into glucose is called gluconeogenesis. The opposite of glycogenolysis is glycogenesis, which is where blood glucose levels are high. We convert glucose to glycogen and store it in our liver. The synthesis of triglycerides from that glucose, that's what we call lipogenesis. Okay. So it's involved with doing all sorts of different things with carbs. It does a lot of different things with fats. The liver cells, hepatocytes can store triglycerides, break them down to generate ATP. That's lipolysis. Synthesize lipoproteins, which are fat protein hybrids. Synthesize cholesterol and use that cholesterol to make bile salts. Something we'll talk more about is deamination, which is the removal of amino groups from amino acids. That can then allow them to be used to make ATP or to be converted to carbs or fats. We'll talk about that in our next lecture. In doing so, we actually form what's called ammonia, which is very toxic to the body, but our liver can convert that to urea, and then it can be peed out. In fact, urea is a pretty big component of our urine. And then previously we talked about our plasma proteins like fibrinogen, the globulins, albumin. That's where they're made in our liver. Our liver processes drugs and hormones, can detoxify things like alcohol, excrete drugs into bile. Can also alter, excrete the thyroid and some steroid hormones. That bilirubin I was just talking about, formed from the breakdown of hemoglobin, is absorbed by our liver, secreted in the bile through a series of different things. That bilirubin, strocoblin kind of are involved and contribute to that brown color that our feces typically is. We've talked about the synthesis of bile salts for the emulsification of fats, storage of things like vitamins and minerals, phagocytosis, we talked about this previously. We have macrophages, specialized cells in our liver that can break down, eat, digest red blood cells, white blood cells, bacteria, and then the activation of our vitamin D along with the skin and kidneys, converting it into the active form, converting and synthesizing, I should say. Very closely associated with our liver is the gallbladder. Is essentially just an extension of the liver, located on in a depression on the posterior surface of our liver. It's connected to our liver by a network of ducts, the direct one being the cystic duct that carries bile from the liver into the gallbladder, where it's stored and concentrated until it needs to be released, and then it contracts because there are contractile fibers pushing that bile down into the common bile duct, and that common bile duct actually joins the pancreatic duct in the same spot, dumping that pancreatic juice and the bile from the bile duct into the duodenum, okay, which is what this figure is kind of showing you here. So overall, the function of your gallbladder is to store and concentrate bile produced by our liver until it's needed in the small intestine. You can get by without a gallbladder. It's a fairly common procedure for people to get it removed. People kind of suffer from gallstones a lot. Very painful, unpleasant experience. People that have a gallbladder, are fine. People that don't have a gallbladder typically are fine. They might have a little more trouble digesting fats because they just don't have as much bile to help with that emulsification. But most people are just fine without. Okay, I'm going to stop here. We will move on to the small intestine, and I will record that lecture next.